Howdy, welcome back to the Idea to Impact podcast. Today, you're going to hear from Dr. Thomas Kent, who works at A&M's IBT campus. And not only are you going to hear Jack and I get schooled on vascular neurology, but you're also going to hear Dr. Kent's story of how research when you are dealing with drugs or with therapeutics may be a little bit different from the guests that we have in the past, especially in terms of how long it can take to get things from the lab to market. So if you are in the chemistry or in the medical field, this is a podcast that you're going to want to listen to because his story is really going to speak to you. So let's get started. Good afternoon, welcome back to the Idea to Impact podcast, where we discuss all things innovation, technology translation, and commercialization. I'm Dr. Beth Duermeyer with the A&M Innovation Partners. I am here with my lovely co-host,
Jack Manhire.
And today we are talking to someone whose path or journey from idea to impact looks a little different from our previous guest, but then again, I think I have said that about every guest,
Right.
with the exception of Mark, because he was our first one. But that is what is so cool about what Jack and I do. Almost no two entrepreneurs or inventors have the same story. So we have, this week we have Dr. Thomas Kent, who is the Robert H. Welch Chair Professor of Texas A&M's Institute of Biosciences and Technology. And we say IBT for short, because that is a mouthful. And they're down in Houston. So you will find out as you listen today that Dr. Kent's story is unique because of the area that he works in. So he is a neurologist, specifically in vascular neurology, and as well as being a teacher and a researcher. And because he's working with drugs, we'll hear that actually that really influences what his story looks like in terms of how long it can take to get an idea from the research all the way to impact. So we're going to get things started. Thank you, Dr. Kent, for being here with us.
Happy to be here.
And why don't you start by telling us how you got into doing research, especially after attending medical school to be a physician. What got you into research?
Well, so as you said, I'm trained as a physician and a neurologist, specifically a vascular neurologist, which means I take care of mostly patients who've had a stroke, and there are other diseases that involve problems with the blood vessels. I also spent several years in basic science laboratories, and so now I do research. First, I trained in a field called basic and clinical pharmacology, which is the study of the way drugs work, and then also how they behave in humans. And then later I trained in a field called electrochemistry, which is how substance reacts with each other by exchanging their electrons. This is a really fundamental process in biology, and so ends up being important in diseases as well. And I also invent things. I invent things that I see there's a need for in the clinic, and use my training and colleagues to come up with a solution. But I didn't go into medicine to do research. My father was a really busy clinician, and I thought that was a great life. But then I was approached actually during my psychiatry rotation by the chairman of psychiatry. I'm still not sure why he picked me. He must have seen something in me that he thought he could exploit. And he passed this really crazy idea by me and pretty much convinced me to participate.
And can you tell us more about this research study that you were recruited to help out with?
So this first study, so it was a really crazy idea. He was very famous in study of alcoholism, mostly in genetics, but he also had other ideas about what was going on. And he had this theory, it's called state-dependent memory, that people that learn something under typically drugs of abuse, including alcohol, actually tend to remember it better if they're intoxicated again. And they also would forget if they sobered up. So his idea was that all these bad things that happen to people while they're intoxicated, they just forget them when they sober up. And so how could he test that? Well, you can test that, he thought, by getting a group of people that have different drinking histories and get them intoxicated, train them something, and then bring them back the next day and either take the test sober or take it with alcohol and see what they do. And so that was the study. And basically it involved recruiting medical students with a range of drinking histories. And it was a little bit surprising what their range was. And then I would, on Friday night, serve them drinks, either a little bit on top as a placebo or a full drink, give them these tests, and wait for them basically to be safe to go home, which is several hours in this testing room with 10, 15 medical students, and then bring them back the next day. And most of them came back the next day. And we did this several times with all the different permutations of the alcohol. And it was just an amazing, incredible experience. I'm
sure you really had to twist their arms to get them to this.
He did not. And there were a lot of volunteers. And as usual, some upset when they got the placebo, but, they quickly figured out, which ones were real and which ones weren't. But it was amazing to see their personalities change. I mean, some became just outright belligerent, others just really pleasant. I caught one trying to do, very odd things to my desk.
And you just have to sit there, serve them drinks, and record, right? You just got to observe and report, right?
Record, tests, actually give them tests, encourage them.
Original reality TV series, right?
I know, right?
Yeah. So anyway, that went on for several months.
Wow.
And when it was over, put the data together, learned statistics, analyzed the results, and sure enough, it was true. They really did remember better from intoxication to intoxication than if they took the test sober. Now, overall, of course, they did worse than if they learned it sober.
Right. So anybody listening to this or watching this, do not think that you'll learn more in school by getting drunk first. That's not what was happening, right?
Now, if we could film this and then go back, and if Dr. Kent could create a time machine and we could go back to like college level Dr. Beth, who, somebody misinterpreted this study a long time before I got to college, but, because we always joke, I'll just, I'm tipsy when I study, I'm just going to be tipsy when I take a test because it works, that's what the science says. So,
well, I hope they didn't use this study for that.
Well, somebody did, because some urban, just know that you're part of some urban legend somewhere.
Yeah, I heard you learn better if you're drunk. Just the sound bite.
Yeah, absolutely. The drunker, the better. Yeah, absolutely.
So that got you into the research part of it? Is that was your first kind of full-on research assignment there?
So that was it. And then they approached me actually to keep doing research. That was the heyday of early days of brain imaging. And at that time, there were a couple psychiatrists, mostly neurologists, but a couple psychiatrists as well, that were working on the actual methods to image the way the brain functions. And it turns out what they're really measuring is the reaction of the blood vessels.
Oh, interesting.
So every organ in the body, when it's exercised or worked, the blood supply to it goes up. That can actually cause some swelling. That's why you rest your arm when it's bruised and swollen, because it'll just get worse if you remove it.
That's true for the brain as well. So if I'm hearing something, is there more blood coming to the part of my brain that is in the oral part of am I thinking about this right?
Absolutely. That's absolutely right. Yeah. And it can, under bad situations, actually steal it from areas if you have marginal blood flow. So this whole thing was discovered. Just an incredible patient had an abnormality of blood vessels called a vascular malformation that has these way too many blood vessels than should be there. And it happened to be in the vision part of his brain, which is in the back. And what brought him to the doctor was that he would open his eyes and he would actually hear whooshing in his head. And he correlated himself, knew something was wrong. The doctor, was a very famous neurosurgeon, actually only one or two neurosurgeons at the time that could even begin to deal with this problem. And sure enough, they made this link between metabolism and blood flow. And right now, that's the phenomenon that we use to measure functional brain imaging. And so this was all just beginning, which led me to wonder how that happens. How is that linked? How are those two things linked? And then that led me to study blood vessels. And then once you're studying blood vessels, you study the abnormalities of blood vessels, and that's stroke. And so that's how I became a stroke pathologist.
So this isn't the first time I've heard a story like this, although I'm not sure if it is at all normal, where you have a patient who, for some reason, either has some abnormality. I don't want to call it a mutation, but some abnormality. But because of that abnormality, medical science is able to make progress. They're able to find something out only because somebody's a little weird in some way, right? If they were just looking at all normal people, they wouldn't necessarily be able to make these connections. Is that kind of right? You see that every once in a while?
That's a really good insight. And so that's this relationship between cause and effect. So when something is normal, especially in humans, you actually have to do something to it to see if it causes something. It's easy to do in the laboratory, but it's harder to do in humans. But then when a person comes to you with something abnormal, that's the abnormality. And so you can make if you're aware enough and observe enough, you can make that link. That's absolutely the way progress can happen. Unfortunate, for the patient,
right?
It's not too serious a condition, but that's absolutely right.
Wow. So some of it's luck. I mean, some of it's just somebody happens to go to the right doctor who happens to know, hey, it's fluid in the ear. Don't worry about it. No, it might be something to do with the vision center of the brain. That's amazing. It's amazing.
And just it always boggles my mind just how many people don't go to the doctor. And so, sometimes there's things that could be going on and we'll just never know about it because people, have fears of going to doctors. And, I'm not one of those people because I have a team of doctors that I like to go through all of my abnormalities. But you mentioned the word stroke, and I think I kind of had like a misunderstanding of what stroke was for a long time. I always heard stroke and I thought I kind of always heard it associated with the word heart attack. So can you tell us a little bit more about strokes and what that is exactly, and what about strokes do you work on most of the time?
Yeah, because just to riff on that a little bit, I always thought, and I think this might be the popular understanding, that if my kids are really stressing me out and I'm on my last nerve, I'm going to start twitching and then part of my face is going to fall down. Is that at all right? Or is that just urban legend?
That's pretty rare.
Okay.
Really bad stress rarely can do that. It probably does it mostly through a super high blood pressure. I mean, it has to be just super high to do that.
Right. So again, very abnormal, right?
Really, off the wall. And that range, and that happens. I mean, it happens so infrequently that when it does, somebody actually reports it. Because it's really not something that happens that often. Yeah, stroke. I know exactly what you mean. I'm so stressed out. I'm having a stroke.
Right.
And that is a common use of the term. But what it actually means is the blood. So the brain has a lot of blood vessels that supply different parts of it. And a stroke is when one of those blood vessels is blocked or it ruptures. Mostly it's blocked, and it's blocked by either a clot that's forming there because you had hardening of the arteries, or it comes from somewhere else. I'm sure you've seen ads for medicines and devices for atrial fibrillation because it comes from the heart and goes right up to the brain. So some of those are just like a heart attack. A heart attack is a plaque where clots form because it's so abnormal in that area. And about a third of strokes are due to that. You heard carotid artery can happen and other vessels. But then about 2/3 of them are other things. And they all get treated differently. We do different things to prevent those. To treat them when you're having a stroke is pretty much the same. You somehow, when it's a blockage, you try to get rid of that blockage, either with medicines or a device that pulls it out. And so that's how we treat stroke now. That's the good news, is it does improve outcome. The bad news is that maybe 10% of people actually get those treatments. Either they get there too late and the brain is just too injured, or they have other problems, or their clot is somewhere that you can't get to it. And so a lot of patients, now some of them get better on their own, fortunately, but stroke is still the leading cause of disability in adults, and way up there in the top 4 in terms of causing death. So it's still a big problem. And so there's a need to find ways to improve the ability of the brain to survive that.
And just so I understand, because I do let's just take the clot, for example. Is the reason that it might cause disability in me if I have a stroke and a clot, because my brain went for too long without blood flow? In other words, if I get the clot out fast enough, I have a better chance of recovering. Or is it one of those things where if I get a clot and I have a stroke, that part of the brain kind of dies, like dying on the vine, and I never will get better? How does that work? What causes the disability, I guess?
Yeah, so a stroke is different than, say, your heart stops. When people giving CPR, then pretty much none of the brain is getting perfusion unless the CPR is good and get in time. You get there and restart the heart. And that way, cells really just have a few minutes. And some survive longer than others, which is why people sometimes can keep breathing after a cardiac arrest. But stroke is a pretty much a single blood vessel. And we're fortunate in a way that we have lots of these blood vessels and they interact with each other. And so for a while, they can actually supply enough blood that your brain can survive. There will be an area that has no blood, and it will, it won't survive, and that's where your disability comes. And then that spreads out. The amount of injured tissue spreads out over time. You can actually see this in models of stroke. You can see that spreading out. So that's why it is time. Time is important for that.
So people who have those strokes that causes that kind of disability, just over time, they're just going to get worse? Is it?
Over the first few hours, once it establishes, they'll get worse because there's some swelling, just like anything that's bruised, there's swelling, and then that actually occasionally can be fatal.
Okay.
And then once that passes, so there'll be a stable thing, and usually happens over the first day or so.
Okay. I thought maybe you said like over time. I wasn't sure what time.
The whole brain becomes dead over
hours. Yeah, I'm thinking more like long term of months to years. So at some point it stops spreading, right?
It stops spreading because that blood vessel will only supply a certain part.
Okay.
And it's that part that if you don't save it, the whole thing will eventually. So it might be a little wedge. It might be a large part, like the whole side. So it depends on which blood vessel and the ability. And so my research is to try to give time to that brain to survive, to find what's called neuroprotection. And there's a lot of people doing this. Up till now, it hasn't worked. There have been a large number of these studies in humans, and pretty much they've all failed. And so, you know, this is an important enough subject that we've taken it on.
But you said it's failed in humans, and I know we're going to get more into this. So that means it's already been through several rounds of animal trials. And so I think you mentioned this in our previous session, but how long typically does it take to get to where you're actually putting things into humans?
So, I mean, it can take 10, 15 years is not unusual to get to humans. And even after all of that, and hundreds of millions, if not, you know, billions of dollars, you get failure, and you get failures after failures. And so people question the whole concept. They question, you know, how we do it.
Right.
And you know, you have to sort of do that. You have to go back and say, well, what did we do wrong, or what, you know, is the idea wrong, and you know, that stuff happens all the time. And I think we can get into more specifics on how that happens, especially.
So is the current state of the science that if I were to have a stroke, and again, I'm generalizing, if I were to have a stroke and I were in the 10% that could be helped, there's some type of device or something you can put up there and grab that clot or fix it or do something with it. And then the 90%, because it's such a large amount, they're the ones that still need people like you to try to find solutions. Am I summarizing it half correctly, at least?
Yeah, it's pretty close.
Okay.
So there's medicines to bust up the clot.
Okay.
A risk of causing bleeding elsewhere. They might actually bust up some good clots and some bleeding elsewhere, but in the end, overall, people benefit. And then there's a slew of new devices. Technology's just been incredible.
Okay.
That can remove that clot. And you may not restore completely. You may still have, you know, that area that had no help from its neighbors, neighbor blood vessels. So you know, hopefully you saved enough that you get better over time. But you know, even then, it doesn't always work. But I've seen just amazing and incredible recoveries with these devices that you would never have seen 5 years ago, 10 years ago.
So the brain's pretty amazing because it has, I don't know if they're necessarily redundant systems the way you're describing them, but that help from the neighbors. And then also it partitions it off that if something's going to die, we're going to make sure it's only a localized spread, right? Am I hearing that correct too?
Yeah.
So that while the part that does die, if it's a critical part, it could be very, very bad for the human.
Right.
But the brain, as it's designed, is trying to save as much as it can if something like that goes wrong, right?
It seems to be doing that.
Okay. Pretty amazing. Wow.
Yeah, we call these actually watersheds. If you watch two sprinklers, you know, they do their own territory.
Yeah.
But they also they overlap.
A little bit. Yeah.
It's that overlap. That's exactly how the brain is designed.
Wow. My brain's a big yard with a bunch of sprinklers. I love it. I can see it now. And so you're at that threshold of trying to figure out what to do next, you know, with the large population that still need help. You know, what are you working on as far as the areas? And I'm sure there's a story behind it too, as far as how you got into it.
Yeah.
But you know, obviously you're making some contributions in this area. What are those?
So the first contribution was actually a way to measure the blood flow and to measure this spread. And that was done with very early days of MRIs in various kind of stroke models. And we actually made the first way to measure blood flow non-invasively with MRI, and combine that with another technique which can measure injury, and we saw that you could see how much blood flow was missing and how much you still had to survive. And that now is actually the gold standard for telling when you come in with a stroke if you're eligible for this treatment.
Wow.
Not everyone has the fancy technology, so now you can use CAT scans to do it and other things to do it, but it's still, compared to this method. I mean, if it's all gone already, then why risk either the drug or the intervention? So that's more on the diagnostic realm, but I use this electrochemistry knowledge to measure something we all know about, and those are free radicals or oxidative stress. We all know about antioxidants.
So I know about those words, but now that you're saying them, I'm not really sure I understand what they are. So free radicals, I picture some type of little things bouncing around and they're doing damage like a video game or something like that. Mm-hmm. But what are those, just for those who aren't sure what they are?
Right. So free radicals can come from a lot of different substances, but the most common one that we deal with is from oxygen. Oh, so okay. The reason oxygen is just such a great thing for life is that it reacts so easily and it exchanges these electrons. But in the process, it actually ends up in a really unstable state. And that unstable state, we have tons of protective enzymes and proteins in our body to protect us from that. It's happening all the time. It's happening all the time. And we're adapted to that. And so it happens, it does its thing, and then something comes along to get rid of it. Gotcha. So we're functioning normally. But as we get older, little bits of damage start accumulating, and people have theorized that's related to aging. And so, that's one of the reasons lots of people take antioxidants. So they're supplementing what your body normally has.
Gotcha.
And sometimes they're the things your body normally uses. You're just taking more of it. Vitamin C, vitamin E, those are things people take.
So your body's in a constant battle with oxygen, even though it needs it. It's constant re-leveling, right? Okay. Right. Gotcha. Okay.
Constant.
It always amazes me what our human body does.
Yeah, absolutely.
It's incredible. Yeah. It is incredible. There's this whole world out there of how we ended up with oxygen and how it's just a really interesting it's a really interesting field in itself, I guess, if you're interested in that. But when you have pathology, and stroke is a great example of that, you overwhelm the thing you have to protect you normally.
Gotcha. They're
just overwhelmed. They can't function. And so this stimulated, so one of my other early inventions was a way to measure it in the living body. So you measure it through electrochemistry, you can measure these radicals coming off. And sure enough, you see these huge bursts, and you see them for a long period of time. You see them, hours and even, a day later, it's still happening. And so, the idea is that that's still there. Maybe you could help even a little bit with some kind of antioxidant. And so there were tons of studies on various antioxidants. They all failed. Every one of them failed. The last one actually was the death knell for big drug companies to do stroke studies, because the estimates are $500 million they spent on this drug.
Wow.
And they thought it looked good, and then it just, when they did the definitive, failed. And so there was, everybody's thinking, well, maybe the whole theory's wrong. Maybe it's not involved. And so what we did is we knew it's there. We knew it's happening, and we just didn't buy that it's not important.
So your theory is still that the body's overwhelmed, and so therefore it's not there to battle and balance out the free radicals from the oxygen intake, right? And that's what you're trying to balance back out? Gotcha. I'm with you. Okay.
And so the bursts of free radicals are, you're seeing those after somebody has a stroke, correct?
Right. You're seeing it both after they have a stroke, and then you're seeing it when you remove the blockage.
Oh.
Now, oxygen is rushing in. You've consumed all your protective enzymes.
Gotcha.
And now, is that really even good? You know?
Right.
Yeah.
So we're convinced, and like we do in research, you have a hypothesis, and our hypothesis was, well, maybe our drugs aren't any good.
Right.
And that's what we reviewed, every single antioxidant that's been tried. And sure enough, when you look at all of them together, they're just not strong enough. They just, I mean, if you had a continuous infusion of gallons of it, you know, maybe then they would have worked, but you know, who can tolerate? That's just able to
expensive.
Right.
Yeah.
So about this time, I was really close at my previous institution to Rice University, and there was a lecture on nanomaterials as antioxidants. And so I attended it, and it was incredible. Now, these are not biologists, these are chemists. And the story of how this came to be is really pretty incredible. But at that talk, these were thought to be antioxidants, you know, just tens of thousands of times more able to do this than any of the small drugs or proteins or anything that anyone had come up with.
And these antioxidants are not naturally occurring; they're like created, or
yes.
Okay.
Right. So
but
this
it's not like Iron Man and Tony Stark, where like little nanobots are doing things, right? Or are you making nanobots, or
there are nanobots now?
Oh, really?
Mm-hmm.
They're really awesome.
Jack's
Best guess yet.
Assembly things. There's nanomachines that can tear up cells. There's nano.
Wow.
Yeah.
Okay.
Yeah.
Is that what you mean by created, though? Is that what you mean by they're created?
They're synthesized, right?
Ah, okay.
Yeah. So this field began. It's actually pretty old field, but mostly in theory. And then a group of scientists, including two at Rice, Smalley and Curl, actually created the first carbon, the smallest carbon material possible. They called them Buckminster fullerenes.
Okay. A lot. Yeah.
Buckminster Fullering was an architect who created the geodesic dome, which is a icosahedral, and it also looked like a soccer ball when you looked at it. Right. So the short is buckyballs.
That's easy to remember. Yes.
The technical term now is fullerene. And this was discovered by actually simulating what might happen in outer space. Just incredible high temperatures under pressure. And then they convincingly showed that carbon could form these circular balls. Thought at the time to be the smallest possible. Now they're probably can be even smaller than that. And they're purely made of carbon, which is what, you know, we're made of.
Right.
So it's not a foreign material.
Right.
And the way that their structure was, you know, what's really cool about chemistry is you can look at something and you can make predictions. And they predicted early on, this is actually in the 90s, that these would be really good antioxidants. And somebody patented it as a treatment and actually patented it for stroke, these fullerenes. So the idea was really good. It didn't progress beyond, as far as we know, the first phase of human studies. We're not even sure it went that far. It's all kind of secret. It didn't happen at Rice. It's all kind of secret, and we're not sure why.
And this was, what, 10, 20 years ago or
It
30
was
years?
in the mid 90s.
Oh, okay.
Mid 90s. So one of the issues may have been that they don't dissolve well in blood, and maybe they stick around in tissue too long. And
Gotcha.
so the Rice chemists set about to design or to develop ones that would, from the same starting material, but would actually probably be more compatible with a human. And that's what I saw. That's the talk that I came into it at that point. And that's what we've done since then. So we took those materials. I can contribute, what design would make them work best, what size they need to be, where they need to distribute. Fortunately, I can look at it too with the chemistry knowledge and sort of understand it to the extent that make that kind of contribution. And also where they're needed, where would we need something like this? And so that's what we're doing. We're working in carbon nanomedicine to develop treatments for various diseases that involve oxidative stress.
So I'm reading a little bit into the story, but if we were going to do the movie about you, and we had the scene where you're at this lecture at Rice, and you start to see what's happening. Is this where the music gets real dramatic and your eyes get really big and you're, Eureka! Like it just all comes together and you go, I know what this can do.
Hovering around.
Yeah, right.
Yeah.
So, beautiful mind.
So, yeah. Yeah, that's exactly what happened. If you look at my notes, they're like, yes, yes, yes.
Yes. That's great.
Yeah,
that's awesome.
That's exactly right. I mean, it was so phenomenal that I actually worried it was too strong because, like I said, you use radicals in normal function.
Right.
So is this going to wipe that out too?
Right. Almost like an antibiotic is going to kill the other bio parts, right? Yeah, right. Okay.
But if anything could work, then I thought these would, and so we do the basic science. NIH funds it. We've done stroke. Traumatic brain injury has a similar problem with radicals, and they work. They work better than anything that I've ever seen. Wow. And they work late. So you can give them, in an animal model of stroke, you can give them at a realistic time when a patient would come.
And the doctor, yeah.
When you look back at some of these failed drugs, they were pre-treating them.
Yes, yes, I see.
They were showing kind of in theory that it works, but not in the sense. And I think that's where being a stroke neurologist, I can dismiss those studies. They're just not going to be, that's not ever going to happen. I mean, yeah, I guess if there's a pill and everybody took it just in case you had a stroke, but you still need pretty high levels. I mean, these things need to be injected. So that's maybe what might happen in the future or preventative. But at this point, that's not realistic. And so they work. They work incredibly well.
And if I understood this, and this might only be one scenario, I understand that, but if you kind of pull the clot out, either with a device or you otherwise dissolve it, is that the point where we'd want your injection to be able to get rid of all the excessive stuff? Is that about right?
That's exactly right.
Okay.
Yeah. You saw that perfectly.
Okay, great.
Right. Because you're now there. And so if you can do the catheter or give the clot busting drug, you can give this drug.
Great.
Because you're there.
Right. You're already.
Yeah.
I like that.
There, you can't give it.
Set up. You're ready to go.
Right.
Yeah.
And you say that it's been very successful, and so that's in animals, correct?
Right.
Okay. So
animals and cells, we do as much cell work as possible. So in cells, we can give it hour or 2 hours later when we give some kind of, injury and so here's the irony, and here's the sad thing, is that it's virtually impossible to find people that will fund the commercialization of a drug for a disease like stroke or traumatic brain injury. After all those failures, to convince people to put, the tens, if not hundreds of $1,000,000 into it, it's just not going to happen. And
is that why? Because of the failures, they've been to Vegas already and they lost, and they said, I'm not going to play that game anymore. Is that it?
yes, sir. Yeah. I mean, that's pretty much it. So there was so much enthusiasm, and not just antioxidant drugs. There's dozens of things that happen in this injured tissue. Molecules are released, some are consumed, lots of different approaches. All of them failed. And so it's not just this. And probably for similar reasons, the drugs just aren't that strong. I mean, this is a pretty overwhelming event. And so that's not a good strategy. And we knew that. I mean, you still talk a little bit, but pretty much, no, you won't find investors, at least in the States. Some smaller countries have expressed some interest, but at least in the States for stroke and trauma.
You would think that with something like stroke that impacts, quite a few number of people, not just in our country, but in the world, you would think that people would want to fix those problems, but because it's failed in the past, people are reluctant, and which to me just sounds completely backwards.
They're reluctant, and what they know now is that these early, they didn't go to these big expensive trials blindly.
Mm-hmm.
They did them in earlier, smaller studies. And maybe a third or more looked promising to them. So the initial amount of money they would spend wasn't in the hundreds of millions. But what happened is then they went to these larger studies and they were negative, and they were probably negative because these diseases are so complex. I mean, no stroke is the same. The patient's different. They may have bad diabetes or may not have diabetes at all. They may have, bad uncontrolled high blood pressure or good controlled. They're so heterogeneous that in a little study, you may just have by chance, a less sick group that got the drug. And so they look like they got better from the drug. And so they know that now, that you're going to have to do a huge study for all these things to balance out. And they're just not going to spend the money at this. So they know even if the early one is positive, it's not going to convince them. It's just not going to convince them to do that.
So is that maybe a contributor to why these things take so long to progress? Is it, instead of, getting one investor or somebody to fund the research, you're kind of having to get some money, do some research, money runs out, find more money, continue the research? Are you just kind of start with the research?
Well, yes, you would definitely stop and mostly start and mostly stop in that scenario. And so what we do now is take a different scenario. We all want to have some beneficial effect for this disease that affects, so many people. Like you said, the impact is so huge, you would think that that there would be good motivation, and there is, but you just have to take a different approach. And so the approach that is most popular now is to do something that the FDA has also realized is a problem, is that diseases that don't affect a lot of people will never generate enough revenue to motivate, or even so that a company can afford to do the research, because they have to recoup that somewhere. And if there are only a few thousand people that have the disease, that's why you see, $1 million drugs when they first come out, because, there were 100 people, it might have cost $1 billion to make that.
Yeah.
So they're called orphan diseases. And so the strategy is to find something that has a similar action and study that. And the FDA actually facilitates it.
Join us next time when Dr. Kent reveals more about the orphan disease financing strategy.